is the one individual in a population that naturally acquired traits that it can withstand the herbicide application. But you won't see that in the initial stages. But with repeated application of same mode of action, which is like same chemistry of herbicide, what happens is the next generation, that weed will survive, reproduce, and that's how the resistance is spread in the next generation. Essentially, it is a consequence of selection. In US, what has happened is over the period of time, the same chemistry of herbicide has been applied over and over again. So now we have seen a lot of cases of resistance to herbicide. So this is a graph where you can see the evolution of resistance in weeds over a period of time. So it's going rapidly, and if you see the last decade, more number of weeds have evolved resistance to herbicide. So if you talk to a grower in the US and ask what is their main concern or main challenge, most of them, more than 80% of farmers say their challenge is controlling the resistant, herbicide-resistant weeds. So that has come to a point there where we are now lack a lot of options to manage these herbicide-resistant weeds. So, and this is a map of US where you see that those numbers indicate how many resistant weeds are present in each state. And I come from Kansas, which is in the center where you see the number 26. That is the Kansas state. And if you look at the numbers, and we rank first in that. We shouldn't be taking credit for that, and obviously I'm not proud of it. You see, we have more number of resistant weeds in our state. That's because the cropping systems in Kansas is very diverse, and the types of weeds we have is also very diverse. So all this combination of genetic factors and external factors result in evolution of resistance. So some of the important weeds in our states are, like I just listed, with a common name and scientific name. Top one, palmer amaranth, belongs to Amaranthaceae family. That's the very, very problematic weeds, followed by kochia, waterhemp, horseweed, and sunflower. These are some of our major weeds. In terms of how these weeds evolve resistance to herbicides is, generally in weed science, we group them into non-target site-based resistance and the target site-based resistance. As the name implies, the non-target site resistance in weeds evolves because of alterations to some mechanisms which have nothing to do with the target. I guess you all know each herbicide has a specific protein as a target in the plant. So the proteins, the herbicide target is still sensitive in this type of mechanism, whereas because of not enough amount of herbicide reaching the herbicide, either because of reduced absorption or translocation, the weed can evolve resistant. But majority of cases you see the metabolism based resistant, wherein the herbicide molecule is degraded or becomes inactive because of certain enzymatic activity, therefore the plant becomes resistant. So the non-target site resistant mechanisms are commonly seen in many weeds for herbicides such as atrazine, or you can even see for ALS inhibitors. Whereas target site resistance, again as the name implies here, the alterations to herbicide target results in the resistance. The target site mutations, especially in the gene coding for that particular protein, we have seen a lot of resistances because of SNPs. A single nucleotide polymorphism results in a change in amino acid, therefore the herbicide cannot bind to the target anymore. The plant becomes resistant. However, more recently we have seen mechanisms such as gene amplification, which is herbicide target gene is amplified several fold. Although the target is still sensitive, but the plant can produce enough enzyme so that even if you apply herbicide, the plant is not going to die. And then we have also seen some cases where increased gene expression without any amplification or modification in the gene sequence can contribute to the increased level of resistance. So in today's talk, I'm going to focus on herbicide resistance via gene amplification in some of the weeds of importance to Kansas. So essentially the gene amplification based resistance has been documented for a widely used herbicide which is glyphosate across the globe. But my colleague in Canada more recently documented gene amplification based resistance to another group of herbicide called ACCase inhibitors. You don't have to worry about that. For today's talk, I'm going to focus on glyphosate resistance based on gene amplification. Okay. Before I go into the details, I just want to give you a brief background how actually glyphosate works in the plant. I'm not sure how many of you have taken a weed science class or how many of you know about this herbicide glyphosate, which is a very widely used herbicide, and it's also a non-selective herbicide, which means any plant that has the shikimic acid pathway actively occurring that can be susceptible to glyphosate. I know, I'm sure you all have taken plant physiology class, and shikimic acid pathway is one of the important pathways in the plant physiology. So the glyphosate as a target in shikimic acid pathway, the target is 5-enol pyruvyl shikimate phosphate. In short, it is called EPSPS. So that is the protein that glyphosate binds to, and as a result, you see the downstream reactions are all inhibited, resulting in lack of essential amino acids such as phenylalanine, tyrosine, and tryptophan. They won't be produced. I'm sure you all know tryptophan is very important for production of auxins. Likewise, all downstream reactions are inhibited and plant will eventually die. That's how glyphosate kills the plant. So like I said, wide adoption to Roundup Ready technology, especially in the Midwestern US, resulted in extensive selection on wheat species. Therefore, we have evolution of resistance to glyphosate in a number of wheats that I listed there. And not necessarily in all wheat species that are listed there have gene amplification based resistance, but majority of them have that mechanism of resistance to glyphosate. So some of the known mechanisms of resistance to glyphosate, again, we have some cases where non-target site resistance has been documented, basically because of reduced translocation, or in some cases like Lolium rigidum, which is a rigid ryegrass, which is a very, very problem weed in Australia. A lot of scientists in Australia, they worked with this weed. They found vacuolar sequestration. What it means is glyphosate is sequestered in vacuole, and the plant is surviving the glyphosate application. So these are some of the non-target mechanisms were documented for glyphosate resistance, but a number of target site mutations resulting in glyphosate resistance have been found in many weeds. In fact, we also have cases where single nucleotide polymorphism resulting resistance, and more recently they have found double mutations and some cases triple mutations resulting in glyphosate resistance. Then, like I said, in many dicot and monocot weeds, gene amplification based resistance is also common. And then in some case, like Poa annua, recently they reported both gene amplification plus mutation in the EPSPS gene conferring resistance. So more and more in-depth research is done. We are able to decipher the novel mechanisms of resistance to glyphosate in wheat species. So again, today I'm going to focus on gene amplification based resistance. Like I said, they have been it's been reported in both dicot and monocot wheats, and in our state, Amaranthus palmeri and Amaranthus tuberculatus are huge problem. Although I highlighted there the Amaranthus spinosus, which is also a closely related species, which is not a big problem in Kansas, but some parts of the US such as Mississippi, it's a huge problem. So my talk is going to focus on those three weeds today. However, in my lab, we are also looking at the mechanisms of gene amplification in monocot weeds that I listed at the bottom. None of those are a problem weeds in US. All these species, they found having resistance come from Australia. I'm collaborating with scientists in Adelaide. They provided me seed. I'm working with these weeds now to look at the gene amplification based resistance. So here is a picture of world map where you see the distribution of glyphosate-resistant weeds. Obviously, you see in North America and South America, they are dominated, and Australia also has quite a bit of glyphosate-resistant weeds, so as in other parts of the country, Europe and Asia also, there is some level of glyphosate-resistant infestation. So, like I said, my talk will be focused on these 3 Amaranthaceae members, and here I show in US where these 3 weed species are distributed. If you see, majority of states in the US, we have Amaranthus A.palmeri or Palmer amaranth, a serious problem, and most of those states also have glyphosate resistance. And the Amaranthus tuberculatus, which is commonly called common water hemp, which is actually distributed in eastern part of the US. If you see those blue stars, it's all distributed in the eastern part of the US. And like I said, Amaranthus spinosus is very predominant in southern part. You see that black star, which is in the state of Mississippi, which is it's not a huge problem, I would say, spinosis, but still a concern to the growers in that part of the US. So these are the fields that are infested by glyphosate-resistant weeds. Can you see soybean at all in that picture? It's all the panicles that you see, or the inflorescences you see there, are all Amaranthus weed. So the thousands and thousands of acres of soybean is infested by glyphosate resistant in US. And if you drive across Midwest, fields looking like this are not uncommon. You have to believe me. Farmers are really concerned about these resistant weeds. On this other side, it is the glyphosate resistant common water hemp distributed in the eastern part of the US. So, gene amplification, I'm sure you would have taken some classes where you learned what exactly is gene amplification, which is basically an increase in number of copies of the gene sequence, right? In a very simple term, that's what it is. Basically, you will have a gene that is amplified by several fold, resulting in increased enzyme produced from that a gene amplification as a product of the gene amplification. But in nature, gene amplification occurs in several organisms, including human beings and microorganisms and plants as well. But how would actually this gene amplification occur, which is during meiosis? I'm sure you all learned about unequal recombination. Because of unequal recombination, mispairing of 2 homologous chromosomes, you end up having one chromatid with a duplicated copy of a gene, one with a deletion. So subsequent meiotic event results in increase in gene copies. And also it can occur because of retrotransposition, which is RNA mediated or transposon based as well. And sometimes in polyploids you see chromosome or entire gene gets duplicated, thereby you see increased copy number. But more recently, we found more cases of extrachromosomal amplification of a gene, resulting in adaptively advantageous traits for plants. So in these 3 weeds that I just mentioned, we have found extrachromosomal amplification resulting in evolution of glyphosate resistance. I'm going to give you very briefly some information on the techniques that we use in my lab to assess the gene amplification, especially for glyphosate resistance. Again, the gene that they target for glyphosate is EPSPS. We did the quantitative PCR. I'm sure many of you may be using this technique. This can measure quantitatively the amplified gene numbers. And we also used a little bit more sophisticated technique called fluorescent in situ hybridization. In other words, it's called FISH, where because of this technique, you could physically see the presence of amplified copies in the genome of the plant. Very briefly, I'm going to go over this FISH technique. Basically, you need to develop a probe with a gene of interest. In our case, it was the EPSPS gene. And then you have to get the mitotic or meiotic stages of chromosome spreads produced either from the anthers or from your root tips, and then you need to have the hybridization of our probe with the specific chromosomal stage that you identified via mitosis or meiosis, and then because these probes are attached with fluorescent tags, once you observe under a fluorescent microscope, you see where exactly this target gene has been hybridized in the genome of the plant. With this technique, what we found was one of the most exciting results so far we have got in our lab, which is we found this extrachromosomal amplification of EPSPS gene in Palmer amaranth species. We published this work last year in PNAS, and we are continuing this work to the next level. But I'm going to share some of the results that have been already published. So the first case of gene amplification based resistance in Palmer amaranth for glyphosate was reported by my colleague at Colorado State University, Todd Gaines. What they found was they just showed that the glyphosate-resistant plant had more number of EPSPS copies distributed randomly throughout the genome. That was the finding of that paper. It was really a novel finding. I really applaud Todd Gaines' work in this area. Because of this paper, what we did was, following that, we had some glyphosate resistance evolution in cancers, and we worked with cancers population. What we did was, if you look at the GS, means glyphosate susceptible plant, this is a metaphase spread. You see the highlighted circles are the two homologous chromosomes showing a very tiny, very faint hybridization signal of EPSPS gene. Obviously, you expect one copy in a sensitive plants on two homologous chromosomes, right? This is a normal wild type here. Whereas in the resistant plants, you see the arrows indicating the hybridization signals of EPSPS gene, which are distributed on almost all the chromosomes, and similar to what Todd Gain was published in 2010. So this is all we said, okay, we got the similar results in cancers population also, basically, which is the random distribution of these copies throughout the genome. But then our collaborators at USDA and Clemson University, Dr. Chris Askey and Bill Mollin, what they did was they took glyphosate sensitive and resistant plants and they developed back libraries containing the EPSPS gene and surrounding genes. So in their publication in BMC Genome, they had about a 300 KB long back library produced, including that particular segment had about 72 other genes which has some significance to withstand abiotic stress such as salinity or drought tolerance. Additionally, it also has an autonomous replicating sequence as well. So this was very intriguing information. So basically, they found some helitron-like structures and also autonomous replication units. That means they have a mechanism by which they can replicate on their own. So then what they did was they extended their size of their back library. When we started collaborating with them, they gave us a back of about 400 KB, which is really, really good for doing fish analysis. Longer the sequence, longer the back, you get excellent signals to do fish analysis. I was really fortunate that they were interested to collaborate with us and they gave us all these backs. Then what we did was, using these backs, we performed fish analysis. So that picture is a meiotic stage of glyphosate-resistant Palmer amaranth, where you see that genome showing hybridization signals on all over the chromatin there, right? But to our surprise, two signals are away from the genome. That's what was intriguing. What is going on? Why these two signals are not associated with any of these chromosomes? Then what we did was, I'm very fortunate to have collaboration with Dr. Bikram Gill. He's world-renowned scientist, wheat geneticist, and they are the pioneers of fish technique. And they are in our university, and I talked to Dr. Gill and they were very excited to work with us. And he has a postdoc. He is an expert in a technique called fiber fish, which is a high resolution fish technique where a DNA is stretched. It's not even chromosome. A single segment of DNA is stretched, and then you can hybridize your probe with your gene of interest. That's what Dal Ho Koo is, their postdoc, is an excellent, he has excellent skill set of doing this fiber fish. Because of his help, when we did the fish analysis with those 6 bags that were provided by our colleagues, to our surprise, what we saw on top, you see a circular form of the structure there. They are alternating bags with red and green color. So we saw the complete circle of that extrachromosomal unit containing this EPSPS gene. So in addition to the circular form, we have also found some linear structures containing the EPSPS gene. So that was like an eureka moment for us. Okay, this EPSPS amplification is occurring extrachromosomally, and also the structure is circular in shape. So it's like a plasmid. Imagine a microorganism with a plasmid, right? It is exactly like that. We never thought a herbicide resistance can evolve via this extrachromosomal element. Really, this was truly an exciting moment. And then what we did was, because we found majority of them were circular in shape, we call them as eccDNA, stands for extrachromosomal circular DNA carrying EPSPS, which is the target site of glyphosate. So then we also, Dalho also was able to do packetine fish, which is a little more higher resolution of fish analysis. These are packetine chromosomes, and if see on the blue colored diagram or picture, you see the dots there, green dots, are all EPSPS signals containing, bags containing EPSPS. And if you closely see, none of those hybridization signals are within the chromatin. They are all attached to the chromosome by some mechanism. None of these circular structures are integrated into the chromosome yet. They are tethered to that chromatin there. So we are now trying to investigate what is the mechanism by which these copies are tethered, just attached. They are not integrated yet. If they are integrated, then you would be able to clearly see them inside the chromatin by fish analysis. So that's the story we had in Palmer amaranth. So the EPSPS gene is amplified extrachromosomally, and that is a circular molecule. So as a next step, we are looking at how these circular molecules are attached to this, what is the mechanism, because along with that EPSPS gene, there are other genes that are important for plant survival under stressful environment like drought or salinity are also getting amplified. Imagine a plant getting so many genes amplified, becoming very robust. It's like a humongous plant. It can survive anywhere. It can take any amount of herbicide, and it can continue to live. So that was story about glyphosate-resistant Palmer amaranth. Now I'll move on to glyphosate-resistant Amaranthus spinosus. You remember that black dot I showed? It was found in the Mississippi state. My colleague Vijay Nandula he identified that weed, and he asked me if we could do some fish work for him. I agreed, and what we did was we used the same probe that we used in Palmer amaranth, and this is the glyphosate sensitive Amaranthus spinosus. This is a wild type which is susceptible to glyphosate. This is the this is the glyphosate resistant Palmer amaranth. And then we got a hybrid, interspecific hybrid, because Amaranthus spinosus and Palmer amaranth, they both have same chromosome number, 34. They are cross compatible. They naturally cross in nature also. They cross easily, and so we got the hybrids. And when we did the fish analysis using the parents and hybrids, again we found the similar mechanism as we saw in the parent Amaranthus palmeri. So basically, the eccDNA, or the circular structure of eccDNA, can transmit across the species. So it can transfer from one species to other and continue to multiply, which is again a kind of scary situation. That means you can spread the resistance between the related species. And then the story of glyphosate-resistant common water hemp or Amaranthus tuberculatus is slightly different than Palmer amaranth. This is the work of my student Andrew Dillon. He did it for his master's thesis. So when he characterized glyphosate resistance in common water hemp, he also did some fish work with this, and then in plants that had about 16 EPSPS copies, what he found was, in addition to 2 homologous chromosomes that had amplified copies, we also found a arrow, not the arrowheads are the homologous chromosomes. The one with an arrow is the one that's an additional chromosome that had amplified copies. So common water hemp is a diploid plant with chromosome number 32. When we counted the chromosomes here in the resistant plant, we found 32 plus 1. Initially we thought it's an aneuploidy or it has an interspecific hybridization coming from some other species. Then our subsequent work, again these two papers were published in Plant Physiology, our subsequent work showed us clearly that the additional chromosome carrying glyphosate target gene is indeed a circular structure carrying these multiple copies of EPSPS gene. So again on your right side you see the mitotic spread where the hybridization signals are shown in green color and red color, right? The hybridization signal that are shown in green color are telomere specific probes. So we developed telomere specific probes and used them along with the EPSPS gene. So when Dalho did this work, you could see only one of that chromosome there shown in arrow did not have any green signals. That means that structure does not have free ends, it doesn't have telomere, so therefore it forms into a circular structure. So at the bottom you can see clearly we show that the extra chromosome is indeed a circular structure, and we call it as extra chromosome ring structure, and then which did not have any telomeres. So basically the story is either it is Amaranthus A.palmeri or Amaranthus tuberculatus. The extra chromosomal elements appear to carry these amplified EPSPS copies, thereby giving plant an advantage to survive herbicide application. Another interesting thing we found in our research was that there is a huge somatic variation in terms of number of copies that the cells have shown. So this is a this is a spread, the prophase spread from a single root tip of a glyphosate resistant Amaranthus A.palmeri. It's a single root tip. You could see clearly each cell showing different number of these EPSPS copies. So at somatic level, you see the variability of the number of genes that are getting amplified. So what we did after this is we took the leaf tissue of a glyphosate resistant plant, leaf tissue from different positions, and then we found indeed they are good. I'm almost done. So similarly, when we did the fish analysis in F1 plants of tuberculosis also, we have seen somaclonal variation in the F1 plants, which means there is cell-to-cell variability in the number of copies of EPSPS gene. So in summary, there is extra chromosomal mediated EPSPS gene amplification in this Amaranthus weed species, which is giving an adaptive advantage for these weeds to evolve and spread resistance. And also along with this herbicide resistant gene, other genes are getting amplified. And this is where I want you guys to think about. Can we take this for this as an advantage for improving crop traits? Can we have crops with a high level of tolerance to the stresses that I just mentioned? So that is some area we want to exploit and see if we could get some genetic information that can be used for crop improvement. The other thing is the stability of these extra chromosomal elements. Some of you may have studied that these extra chromosomal elements are not stable. Once you take off the selection, there is a potential that we can make these plants susceptible. That's what is our goal also. Let this glyphosate-resistant plant grow in isolation, don't apply any herbicide, and then see if we can make them susceptible. So with that, I'd like to thank my collaborators on the top picture there on left-hand side. The person with a hat is Dr. Gill. He is the key person for my work on fish analysis. His group is, like I said, pioneers in fish techniques, and we worked with them for this. And then on the right side, Dr. Bill Molin is from USDA, ARS, and Chris Askey is at Clemson, and the below bottom is my group at Kansas State University. Without their support, obviously, we can't do any good work. Really thankful to all of those collaborations and students, and also my funding agencies. With that, I'll take any questions if you have.
The discussions with ma'am can be done during the tea break, please. Thank you, ma'am, for sharing a part of your research work with us here today. May I call upon Dr. S. Geeta, Director, CPPG, to honor the guest speaker with a memento.